in this presentation, we will be moving through of the different parts of a cell, especially eukaryotic cell. Remember the difference of eukaryotic and prokaryotic. Some organelles and their function in a cell. So, and also some of the processes then. Okay? So, we know that all organisms are made up of cell. Whatever organism is that, it's made up of cell. So, this is one unity that we can get from all organisms or from life, diba? So, last examination we talked about, I asked you to define or describe unity in diversity of life. So, this is one unity. So, regardless of what kind of organism you are, so we are all made up of cell. Okay po? So, that unites us all. Okay po? So, the cell is the simplest collection of matter that can live. Okay, so cell are living organisms or living things are composed of cells. Okay po? So that excludes the non-living organisms. So cell structure is correlated to cellular function. So whatever will be the structure, that's gonna be the function. So it's correlated. Hindi yung the structure is wings, but then yung function is for swimming. So that's different. Whatever will be the structure, that defines the function of that structure. Okay, so ito yung this is one thing about evolution to talk about. All cells are related by their descent from earlier cells. Okay, so diba, one of the cell theory that is present is yan, all cells came from pre-existing cells. Okay, kasi we are all related from our ancestor. We are just descendants then, and these characteristics of descendants are acquired or inherited from our ancestors or from the ancestor cell then. Right? So, yan. So, here is a typical illustration of a cell. Okay? So, this is a good illustration or a graph of a typical cell then. Okay po? So, later on in these slides, we will be talking at how can we capture cell like this? Diba sabi nga, sir, a cell is the smallest living unit of life, and we cannot see it by unaided eye. So we use different devices, technologies to actually get a picture or capture of a cell like this one. So we use different kinds of microscope, electron microscope. Under this there are many more, compound light microscope, which we usually use then, right? Alright, next. Okay, so though usually too small to be seen by the unaided eye, cells can be complex. Okay? Complex, especially eukaryotic cells, in a way that they have parts, they have organelles which perform a particular function for a cell to become useful, for a cell to become functional in our body. Okay pa? So, yan, cells can be complex then, especially eukaryotic. Okay, so scientists use microscope to visualize cells too small to see with the naked eye. So, diba, that's the tool we have right now. Okay, so we have one kind of microscope that we usually use, and that is light microscope. Talking about light microscope, we use visible light. Visible light that passes through a specimen and then through glass lenses which magnify the image. So this is what we use. So light microscope can be two of a kind. We have simple light microscope, or yung tinatawag natin usually na magnifying glass. You call that class as simple or single lens microscope, meaning isa lang yung lens niya. And the other kind of light microscope, we have compound light microscope. Compound light microscope uses two or more lenses to magnify a specimen. Specimen is the subject under study, say for example a cell. Okay, compound light microscope uses two or more lenses. Diba sa compound light microscope, what are the two lenses there? Can you give me the two lenses of compound light microscope? Anyone? You can participate in a chat para naman meron kayong participation. Anyone?
Objective lens and eye lens.
Diba? So we have two. So very good for someone who answered. We have objective lens. Diba? Objective lens, we have three. We have oil immersion objective. We have tatlo yan. Ano yung tatlong objective lenses? Yung iniikot-ikot natin using a revolving nose piece. Yung isa, OIO, oil immersion objective lens, which can magnify the most. And the other one, we have the yung maiksi, short objective lens, and the other one is the large, yung mas malaki, long. I don't know, I forgot the names of the three, but then yan, tatlo yan. So, and the other one, the other lens, aside from objective lens, we have yun, so yung sabi nga ng isa dito, we have eye lens, or we call this class as ocular lens. Ocular, eye, parehas lang yan. Or sometimes you call it as the part of microscope, we have eyepiece. Diba? So we use two lenses to magnify a particular specimen din. Okay, but then compound light microscope can better magnify than simple microscope. Yung magnifying glass na tinatawag natin. Okay? So, there are three quality of image where microscope can depend on. So, one is magnification, the other is resolution and contrast. So, let's talk about magnification. Magnification is the ratio of an object's image size to its real size. Ano ba yung size niya after magnification? Ano ba yung real size niya? Can it magnify 10 times the size of an image, di ba? Or 10 times the size of a specimen? 10 times, meron niyang 40 or 50 times, meron niyang 100 times, di ba? Yung sa compound light microscope. So that is magnification, the ratio of object's real size and magnified size, or sometimes tinatawag natin to class ng image size kasi hindi na siya yung actual subject na yan, image. Yan. Anyway, next we have resolution. Resolution is the measure of the clarity of image. Okay, clarity. Talking about clarity, we talk about clearness. Gaano ba kalinaw? Kasi there are pictures na masyadong magnified but then not clear. So, we should increase the solution to make it clear, or the minimum distance of two distinguishable points, 'di ba? In a picture, 'di ba? Kapag pag mababa yung resolution, masyadong pixelated, okay? Masyadong mataas yung distance between two pixels, right? So, pero pag mataas yung resolution, say for example, HD, HD, or let's say for example 1280 by 720 pixels. So medyo mababa yung yung distance. Pag mababa yung distance between distinguishable pixels, then the resolution is said to be higher. Right? So resolution has something to do with the clarity of the image. Okay po. So, the higher the resolution, the higher the clarity of the image. So again, another quality that we talk here is the contrast. Contrast is one of the, say for example, one of the tool in editing pictures like that. So when we talk about contrast, it pertains to the visible differences in parts of the sample. Visible differences in parts of the sample, diba? So, talking about contrast, the contrast of light and dark color in a sample, diba? Say for example, the background is white and the picture itself is, say for example, blue. Meaning, kapag sinabi natin contrast, the whitest possible background and the bluest possible foreground, or the bluest possible image of the picture. So, visible differences. You can differentiate between colors of the image. Okay, so nakikita natin na, diba? So, say for example, you take a picture of yourself, you can distinguish which one is your skin and which one is your shirt because of higher contrast, diba? Pag very low yung contrast, you can not actually distinguish parts of your body then. So, pagkakamalan mo na yung t-shirt mo, yun pala yung body part mo. Okay po? So, the higher the contrast, the higher the visibility of the picture. Gaano ba ka-visible yung parts of a picture then. I hope you can get this one. Alright? So, here is an example scaling diagram. Tinatawag natin to class na scaling diagram of the different organelles of the different objects, then scaled objects that a microscope can see, or our eye microscope and electron microscope. Kasi, diba? So, unaided eye, we don't use microscope. Okay, so light microscope and electron microscope, which is the highly magnified microscope that we have today. Okay, so unaided eye, we can see down to a frog's egg, diba? Up to less than a millimeter object or size of an object. We can see humans. We can still see nerve cells or muscle cells by our unaided eye. Chicken egg. Okay, so yan. Yan yung length or approximation of the scale that our eyes can see. But then, less than a millimeter, we have here micrometer. About micrometer, we use microscope then. Okay, so, diba, ang milli, that's 10 raised to negative 3. Ang micro, I think that's 10 raised to negative 6, I think. Okay po. Basta ang micro, nano, 10 raised to negative 9. Okay, so pababaw ng meter yan. Okay po. So, kasi yung millimeter, it's the thousand part. 1,000 merong TH of 1,000 of a meter. Okay po. So, yan. So, we have here the plant cells and animal cells. So, between 10 micro to 100 micro. Kasi, cells can be of different sizes depending on what kind of cell is that. Okay po. So, it's between 10 to 100 micrometers. Okay, so the smallest possible unit of all things, we have the atoms. Okay? So, that can still be observed. That can still be seen using highly graded microscope. Okay po. So, yan. So, tignan nyo na lang yung yung scaling method na ginamit. So, screenshot nyo na lang siya if ever you want to explore more about this. Okay? So, light microscopes can magnify effectively to about 1,000 times. So, imagine nyo na lang na you are the smallest possible thing on Earth. Then you are magnified 1,000 times. Imagine kung gaano ka nakalaki. Okay pa? So, yan. Various techniques enhance contrast and enable cell components to be stained or labeled. Kasi, sabi ko nga, even though you can magnify cells effectively, mataas yung resolution ng microscope mo, mataas yung magnification, but then kapag yung contrast ay mababa, so you cannot actually distinguish different parts of the cell. Say for example, hindi mo ma-distinguish which one is the organelle which one is the mitochondrion, which one is the chloroplast if mababa yung contrast. So, one way to increase the contrast of particular subject like cell is to stain the subject or label the subject. Okay, so kaya gumagamit tayo dito ng mga staining techniques. Gram negative, gram positive. So bacteria are actually based from staining techniques. Okay? Most subcellular structures, ito na yung sa eukaryotic na sinasabi ko, or yung mga sa loob ng cells. Ano ba yung structure sa loob ng cell? Including organelles ang pag sinabi kasi natin class na organelles, it's a membrane enclosed compartments. Merong compartments. Pwede nating tawagin na yung mitochondrion, it's an organelle, nucleus, it's an organelle. Kasi they are membrane enclosed. They are separated from other organelles using a membrane. They are too small to be resolved by light microscope. Kasi if you're going to look at this one, okay, so tignan nyo yung light microscope. That can just see down to mitochondrion, di ba? Hanggang mitochondrion lang yung nakikita niya. Beyond that, yung mga mas maliliit pa sa mitochondrion, hindi na kayang gamitan ng light microscope. Gumagamit na tayo ng electron microscope, di ba? So, yung ribosome, hindi na yan kayang tignan ng light microscope, ribosome, proteins, but then we use then electron microscope. Okay? So here are the different staining techniques in order for us to assign a particular contrast to an image after magnification. So here are the different techniques or contrast techniques. So we have unstained specimen. So you call this one as bright field unstained specimen. You're going to compare that with a stained specimen. So see, you will probably have a greater difference between parts of the cell being viewed in an image. Okay po? So that is around 50 micrometer. Alright? So, phase contrast, mas mataas yung kanyang contrast compared to a bright field specimen. So, kung makikita nyo talaga, you can actually distinguish which one is the darkest, which one is the lightest. Diba? So, yan. Yan. Ito, mababa yung contrast niya. Medyo nagbi-blend yung parts to yung foreground, nagbe-blend yung foreground sa background. Diba? So ito, you can actually distinguish. The difference is higher between parts. Okay, so here is another differential interference contrast. We call this one as Nomarski, diba? So meron niyang filter din sa mga phones niyo. If you try this one, probably you can actually notice bumps on your face, diba? Like pimples, diba? So, Yan yung sa differential interface. You can differentiate yung bumps, yung sa foreground and the background. Kaya medyo may pagka 3D looking. Okay, here is a fluorescence technique. Yung pinakita kong image kaninyo sa inyo at the first or second slide. So, di ba? So, it uses fluorescence technique. Fluorescence, meron siyang the magnified, the image contain different colors, kaya nga fluorescence, okay, of its parts. Ito naman yung sa confocal. Anyway, so yan. So there are two basic types of EMs or electron microscope to study subcellular structure. So here we are going to focus on subcellular structure. Sabi ko nga, our coverage for our exam will be only about parts of the cell. Okay? So also yung pag-uusapan natin dito kasi may mga added topics. So we have here scanning electron microscope. So abbreviated as SEMs. Focus a beam of electrons onto a surface of specimen, providing images that look like 3D ones. So, scanning. So, pag sinabing scanning, yung surface lang yung kaya mong ibigyan ng image. Say, for example, yung cell, yung surface lang ng, say, for example, surface ng mitochondrion, surface ng nucleus. So, 3D image. Pag sinabi naman natin class na transmission electron, from the word itself, transmit, it can pass through the specimen. Okay, it can pass through the internals of mitochondrion, it can pass through the internals of nucleus. So kasi nga transmit. Okay, so tandaan po yung pinagkaiba ng scanning sa transmitting or transmission. TMs. So, TEMs are used mainly to study internal structure of the cell. Kasi, sabi ko nga, kung gumamit ka lang ng electron microscope, ang kaya lang niyang i-study, scanning electron microscope is yung surface of the cell. Okay, ano ba yung composition ng surface of the cell? But it cannot penetrate what's inside. So, we use here TEMs. So, this is used mainly to study the internal. Kumbaga, internal organs ng cell. Are you following? Are you still here?
Nakakala pa po.
Ano
Yes.
po? Very good. Okay, so follow na lang kayo. Okay po? Medyo low energy si sir nyo ngayon. Kasi nga sabi ko, I'm sick. But then, ang gusto ko na lang sa inyo is makinig. Okay po? Okay, so next, kasi I have to reserve my energy for another class later in the afternoon. Okay, so next, okay, so here are the differences. Here are two images of the two. We have SEM and TEM, diba? So, talking about SEM, you just produce a 3D image of what is the cilia look like. Cilia is particular parts of a cell, or it's a form of, anong tawag ito? It's a cell. Okay, so capable of wave-like motion. Okay, so meron yan sa ano natin, sa throat natin to expel bacteria. Okay po. Meron yan sa intestine natin, capable of absorbing nutrients. Sa small intestine, present ang cilia. So, if you look at these two images, you'll see the difference, diba? So, one is providing a 3D images or 3D image of what is the subject look like on the outer side. Yung TEM naman, it's what's inside, diba? Kasi it can penetrate. Okay, so we have this term class self-fractionation. Pag sinabi natin class na self-fractionation, diba? You know what fraction is? It's a part of a whole, diba? So that's the simplest definition we can give to a fraction, part of a whole. So cell fractionation takes cells apart and separates the major organelles from one another. So, diba, in a single cell, we get the mitochondrion, we get the nucleus, the Golgi body, endoplasmic reticulum. We fractionate it. Okay, so to better understand each function. Okay. In cell fractionation, we use the process ultracentrifugation. Okay, so ang ultracentrifugation, if we can decipher the word from itself, pag sinabing ultra, most, higher, diba? Pag centri, we're talking about circle here, diba? Centrifugal. Pag sinabing fugal, it's moving away from the center. Diba? So, pag sinabi natin centripetal, it's moving towards the center of the circle. So, here we use ultracentrifuges fractionate cells into their component parts. Diba? So, how can you better understand the definition of ultracentrifugation or ultracentrifuge? Say for example, your nagtitimpla kayo ng kape, 'di ba? We have in circles or in rotational forces, we have two kinds, 'di ba? We have centrifugal and centripetal forces. 'Di ba napag-aralan niyo siya sa grade 8 nyo? 'Di ba no first month, 'di ba? Different types of forces. Pag sinabing centrifugal, in a revolving body or rotating body, parts tend to move away from the center. Okay? So for example nga, nagkakape kayo, 'di ba? If you stir the coffee then, so water molecules may stick to the edge part of the mug. Okay? So because of fugal force, 'di ba? Nag-stick siya sa mug. Okay? So medyo paano siya. Centripetal force is the force in a rotating body or revolving body where parts tend to move to the center. Say for example, one application of this can be the whirlpool, 'di ba? In a whirlpool, if you will be in a big whirlpool, the tendency is your body will be then attracted to the center of the whirlpool. Okay, so that is centripetal force. Okay, so yan, centrifugal, tandaan nyo na lang yun yung focus natin. So parts tend to move away from the center. Okay pa? So yan, so we fractionate cells or organelles of the cell using centrifugal, ultracentrifuge. Mabilis na pag-rotate. It enables scientists to determine the functions of organelles. Bakit? Yan, sabi ko nga, isolating parts of the body will determine the function of it. Okay po? So, also, with the help of biochemistry and cytology, we can correlate cell function and structure. Diba? Sabi ko nga, at the start of this discussion, whatever will be the structure that defines its function. So, here is a typical centrifugation technique. So, kung makikita nyo, ito yung centrifuge apparatus. Diba? Makikita nyo, we have test tube on both sides of the on both sides of the yan, yung apparatus. Makikita nyo, okay? So, it will rotate on its own very, very quickly. Okay po. So, yan. So, we use the term here, times gravity. Okay, so kung makikita nyo, here, the very first start, we have tissue cells. Say, for example, a blood cell. We get a sample from our blood. Okay po. Then, after that, we need to homogenize. Pag sinabing homogenize, we are going to break the cell membranes of the cell using a particular solution. Okay po, we're going to break that in order for the cells or tissue cells to homogenize. Diba? In grade 7, pag sinabing homogeneous mixture, So, nagiging one phase na lang siya. Okay, you're going to mix it very well. Then, after homogenization, you're going to place it in a centrifugal apparatus. So, like this one. It will rotate on its own very, very fast to the way that larger molecules will be under centrifugal force. It will move away from the center. Ano yung center? Ito. Okay? So, here, at 100 times the force of gravity for about 10 minutes, you're going to separate pellet-rich in nuclei and cellular debris from the homogenate. Diba? So, we separated using differential centrifugation. Pag sinabing differential, we can differentiate which one is homogenate, which one or the pellet-rich. Okay, so, eto na. We can now separate the pellet-rich in nuclei and cellular debris. Okay, and yung nasa taas still, you call this one as supernatant. Okay, they are not affected using this 1,000 times the force of gravity for 10 minutes. Then, we separate the supernatant into another tube. Okay, then we centrifugate that for the data of 20,000 times the force of gravity for 20 minutes. So, it times 2. Times 2 yung number of time, tapos times 20 naman yung force of gravity. So, by this, you can actually separate pellet-rich in mitochondria and chloroplast if cells are from plant. So, we can separate na. Kasi, largest one is the nuclei. Next, we have mitochondria. Pinakamalaking part sa cell is nucleus. Next, we have yung mitochondria. And na-separate na natin yung nucleus. Okay, then the supernatant will be then transferred to another tube and will be centrifugated for about 80,000 times the force of gravity for 60 minutes. For 1 hour, siyang umiikot-ikot. So, here we can separate pellet-rich in microsomes. These are pieces of plasma membranes and cells internal membranes. Membranes which are already broken down into pieces, yung mga maliliit. Kasi pinag-homogenize na natin siya. Okay, then after that, it will be transferred. We can get for 450,000 times the force of gravity for 3 hours. We can get the ribosomes, which are the smallest part of a cell. Pellet-rich in ribosome. Pinakamaliit siya. Nagigets niyo yung sa illustration? Nakuha? Okay, very good. Medyo sinisip mo talaga ako. Okay. Ito pala yung mas pinalaki. Okay, so we homogenize. Okay, so after that, we centrifugate. So, alam na. Discuss ko na siya. Okay. The basic structural and functional unit of every organism can fall under 2 types of cell. We have prokaryotic and eukaryotic. So, alam na natin to. Prokaryotic can be single-celled organisms. These are single-celled organisms. They can live on their own with only one cell. Okay po. So, bacteria, archaeans, diba? Eukaryotic, on the other hand, these are cells which contain nucleus and other membrane-bound organelles. Here, we focus on the eukaryotic. Okay po. So, yung mga parts ng cell. Only organisms of the domains bacteria and archaea consist of prokaryotic. Ito yung sabi ko. On the other hand, protists, isama na natin yung protist, though protist is still a confusing kingdom in the kingdom. For the sake of this discussion, let's say for example, hindi pa na aayos yung issue sa protist. Protist, fungi, animals, and plants consist of all eukaryotic cells, meaning either one-celled or multicellular cell. They are eukaryotic. They are composed of internal organs. Okay? Or organelles. Okay, so Basic features of all cell, no matter prokaryotic and eukaryotic, they contain plasma membrane. Plasma membrane can be termed as cell membrane. We have cytosol, or the semi-fluid substance. Sometimes we call this one as cytoplasm, but then it's cytosol, semi-fluid substance. This is where parts of the cell are suspended on, diba? So para lang siyang semi-fluid jelly-like substance. Okay? So doon nakasuspend yung organelle like nucleus, mitochondria, and what more. Okay? Chromosomes, on the other hand, can be found in prokaryotic inside the nucleus. So they carry genes, they carry the DNA. Okay? So ribosomes, on the other hand, is capable of making proteins. Ribosomes are then made inside the nucleus, particularly sa nucleolus. Then capable siya sa paggawa ng protein which our body needs. Diba? Yung sa RNA. Okay, so prokaryotic cells are characterized by having by not having nucleus. Wala siyang DNA inside the nucleus, but then lang siya, the DNA naka-in place lang siya sa region na tinatawag nating nucleoid. Ang nucleoid is just a region, hindi siya class compartment. Kasi kung compartment siya, ang tawag sa kanya nucleus. Okay, so you call that class as nucleoid. This is where the DNA of a prokaryotic cell is suspended on. So, no membrane-bound organelles like mitochondria, chloroplast, Golgi body. Cytoplasm bound by the plasma membrane. Okay pa. So, yeah. So, here is a typical illustration of a prokaryotic cell and their parts then. So, you'll be able to notice the difference between the image, diba? This one is an image typically produced by TEM, and this one is animated image na lang, okay, or tinatawag natin digitally produced image or digitally enhanced image. So yan, you will see different parts. Okay, ito na yung nucleoid, so ganyan lang yung itsura niya. This one is a rod-shaped bacteria, right? Rod-shaped, or the bacillus. Okay, so nucleoid, comparing that with that of the real image produced by TM. We have ribosomes, yung mga dot na yan, yung plasma membrane, cell wall. Okay, prokaryotic cells contain cell wall, just like plants. They are for the protection. We have the capsule as another layer for a bacterium. Flagella for movement. Okay, so para siyang pang-swing. Pang-swimming ng bacterium. So that's the typical illustration of a bacterium. Okay, eukaryotic cells, on the other hand, contain a DNA bounded by a nuclear envelope. You call this one as nucleus. So meron siyang organelles, yung cytoplasm niya, cytoplasm in the region between the plasma membrane and nucleus, diba? So, kung sa eukaryotic, yung cytoplasm nakabound lang siya sa plasma membrane. But since in a eukaryotic we have nucleus, cytoplasm is bounded between a plasma membrane and nucleus. Okay, so next, eukaryotic cells are generally much larger than prokaryotic. So, ito yung tatandaan nyo. The more complex you are, the larger you can be. Simpler you are, the smallest you can be. Okay? Talking about plasma membrane, or ano yung tawag dito, cell membrane. It's a selective barrier. Pag sinabing selective, it can only choose who can passively enter or exit the cell. Okay? Selective barrier that allows sufficient passage of oxygen, nutrients, waste to service the volume of every cell. It looks like a gate for a typical cell. Siya yung gate where the inside lot can serve as the internal of the cell, and the outside lot can be served as the external of the cell. Plasma membrane, pag sinabing selective barrier, siya yung nagdidesisyon kung sino ba, kung ano ba yung papasok sa lote mo. Okay, parang meron siyang guard, okay? Ito, VIP, papasok siya. Siya yung maunang papasok. Meron siyang daanan. Ito, hindi siya VIP, mahuhuli siya. So, parang ano. Okay, so, typically called as the gate of the cell or the passageway ba. Membrane is a double layer of phospholipid. Okay, from the word itself, phospholipid containing phosphorus and lipid structure. Double layer siya, class. So, in a phospholipid, dalawa yan. Dalawang region ang sa phospholipid. We have hydrophobic, meaning hydrophobia. Okay, so water-fearing region of the cell membrane. Okay, so we have hydrophilic region. So, this is the water-loving. Diba? Water-loving region of the plasma membrane. So, ito yung phospholipid, contain siya ng dalawa. We have head and the tail. So, we call this one as hydrophobic tails and hydrophilic heads. Yung mga ulo gusto sa mga basa, sa mga tubig. Yung mga buntot ayaw sa basa, ayaw sa tubig. Okay, so ganyan yung structure niya. It's upside down. Okay? So, this one on the upper portion of the plasma membrane is the environment outside the cell. Ito naman yung inside the cell or internal environment of the cell. As you can see, there are embedded proteins there. So, proteins allow the entrance and exit of a particular material in the cell. Okay, so, or outside the cell. So, here are proteins. Yan, so meron siya. Proteins, there has to be an attached carbohydrate side chain na tinatawag. So, ganyan yung kanyang structure. This one is the TAM produced, sabi ko nga TAM yung ginagamit natin. So, this is the outside cell, inside cell. Okay, kung makikita nyo, double layer. Dalawa yung layer niya. Dalawa yung ulo, baba tsaka taas, tapos dalawa yung tails sa gitna. Okay po? Yan. The logistics of carrying out cell metabolism sets limits on the size of cell. So, let's skip this one. Hindi natin siya pag-usapan. Okay, so the eukaryotic cell has internal membranes that partition the cell into organelles. Okay, so plant and animal cells have most of the same organelles, but then merong pagkakaiba, di ba? Ang plant, merong cell wall, chloroplast, ang animal wala. So, this is an extra task for everyone. I want you to, talking about the parts of the cell, I want you to give the difference of cell, ano ba yung pagkakaiba ng plant and animal cell. So, this is an extra task for you, okay? So, make a research on, anong tawag dito? Make a research on the difference of plant and animal cells, especially talking about its parts. Okay po? So, yan. This one is a typical animal cell structure. So, ito, this is a structure where all-in-one na siya. Pag sinabing all-in-one, all of their parts in one illustration. So, kaya, meron na siyang flagellum isa, flagella dalawa. So, yan. So, paki-screenshot po para at least alam nyo. Okay, so this is an animal cell. All-in-one na siya. Okay po? So, I am quite pretty sure na pinag-aralan nyo na siya given we don't meet for about 2 meetings. So, napag-aralan nyo na siya yung the different parts of the cell. Kasi binigay ko nga siya sa inyo as an assignment to work on. Okay po, paki-screenshot na lang po. And this is a plant cell then. Paki-screenshot so that you'll be able to study it. Comparing the difference of a plant and animal cell, talking about its parts. Okay? Done. Okay, so let's talk. Okay, so nucleus contains most of the DNA of eukaryotic cell. So it's usually the largest part of the cell, nucleus. So that's there, okay, for plant, and we have for animal. So nucleus contains three. We have nuclear envelope. It's the envelope that covers the nucleus. Okay, so nucleolus. Okay, it's inside the nucleus where ribosomes are made, and the chromatin, so or the DNA or the genetic material. Yan. Okay, nuclear envelope, nucleus, and chromatin. Ribosomes, which are then made from nucleolus inside the nucleus. It goes out of the nucleus then. Okay, and they need to carry the information from the DNA to make proteins. Okay, so proteins are important in our body functions. All the bodily functions are governed by proteins. Different kinds of proteins then. Okay, nucleus contains most of the cell genes and usually the most conspicuous organelle. Pag sinabing conspicuous, parang nakakapagtaka na yon, because this contain class the DNA. Yung pinag-aaral natin, or pag sinabing DNA kasi class, it's the blueprint of life. It's the instruction where ano ba yung life, ano ba yung magiging, or ano ba yung capability niya, ano ba yung magagawa niya. Okay, it's all encoded in the DNA. Nuclear envelope encloses the nucleus, separating it from the cytoplasm. Kaya sabi ko nga, cytoplasm is bounded by the nuclear envelope and the plasma membrane. Nuclear membrane, yung membrane ng nucleus, it's also a double membrane, same as the plasma membrane, and it consists of lipid bilayer. So, hindi na siya phospholipid, but it's a lipid bilayer. So, ganyan. So, this is an illustration showing you the nucleus. Okay, so, here is the nucleus. Around it, meron siyang rough ER, which contain dots. Yung mga dots na yan, these are ribosomes. Pag sinabing rough, they contain ribosomes attached to it. Okay, so yan. So, nuclear. So, this is the nucleus. We have nucleolus. We have the chromatin. Para siyang mga ugat-ugat, diba? So, ang chromatin, it's a tangled form of DNA. Diba? Nuclear pore. If you're going to magnify this part, you'll see that here, diba? Meron siyang nuclear pore. Nuclear pore opens when a material has to enter or exit the nucleus then. Nuclear pore contains of a complex of proteins. Okay, so ganyan po siya. Alright? Ito naman yung sa surface of the nuclear envelope. So, ano yung ginamit niya dito, class? Pag gumamit ka ng surface, you use SEM or scanning electron microscope. Okay, so kung makita nyo, you can see the pores, you can see the ribosomes, mga dot na yan. Ito naman yung TEM. Yung sa TEM, so ganyan. So may mga structure. We have another term, nuclear lamina. Okay, so it looks like a fiber then. Yan. So, inside the nucleus, you call this as lamina. Para siya mga fiber-made parts. Pores regulate the entry and exit of molecules from the nucleus, in and out. The shape of the nucleus is maintained by nuclear lamina. Para siyang cytoskeleton. Okay? Kaya fiber. So, that maintains the shape and structure of the nucleus, which is composed of protein. So, they are composed of protein, mga fibers. Okay pa? In nucleus, DNA and proteins form genetic material called chromatin. Chromatin class, it's a mixture of DNA and proteins. Okay po. So, kaya dito na sinabi DNA and proteins kasi meron siyang a DNA wrapped around a particular protein called histone. So, that made up the chromatin. Okay pa. So, chromatin condenses to form discrete chromosomes. Ito na yung mga nakikita nyo sa libro, sa YouTube, yung chromosomes na X-shape, di ba? Yung X-shape, these are chromosomes which are a condensed form of chromatin. Nag-condense lang siya, parang kumbaga linamig. So, and nag-condense. So, yung mga X-shape, di ba? We have actually, in humans, we have actually 46 number of chromosomes including the sex cells or sex chromosomes then. Nucleolus is located within the nucleus and is site of ribosomal RNA or rRNA. Pag sinabing rRNA, ribosomal ribonucleic acid. Okay po, ribonucleic acid. Pag DNA, deoxyribonucleic acid. Ribosomes, hindi siya ribosomes. If you get the components of it, it's composed of both RNA and protein. They carry out full protein synthesis in two locations. So, later, probably next month or next month, we will be talking about protein synthesis. How we actually made protein out of cellular processes. Diba? Yung sa genetic expression. So, how can we express our genes into proteins? Okay, so two locations. Ribosomes can be found in the cytosols. You call this class as freely moving ribosomes because they can freely move. Okay, cytosol or in a cytoplasm. On the outside of the endoplasmic reticulum or nuclear envelope, so makikita mo class yung mga bound ribosomes, yung mga naka-attach, nakadikit, something like this. So may mga nakadikit, you call that class as bound ribosomes. But then sabi ko nga, yung ribosomes, they are capable of protein synthesis, they contribute in protein synthesis, and is made in the nucleolus. Lumalabas lang sila ng nucleus after maturity. Okay pa? So bound ribosomes, naka-attach sa ER, pwede namang sa nuclear envelope. Ito yung nuclear envelope. Ito yung ER. Typically, rough ER is the type of ER which ribosomes attach to it. Pag smooth ER, konti lang yung ribosomes na naka-attach or minsan nga wala. Okay pa? So, yan. Here one is the cytosol or the cytoplasm. Here is the ER of the cell. So, yung kaninang pinakita ko ito. Yan. Free ribosomes, siya yung mga nakasuspend sa cytosol. Bound ribosomes, ito yung ER kasi. Ang ER kasi class, ganito yung itsura niya. It's folded like, madami siyang mga infolded. Okay, so meron siyang space in between. Meron siyang surface din. So, kung makikita nyo, free ribosomes, wala siya sa surface. Yung mga nasa surface, you call that as bound ribosomes. Ribosomes, if you're going to look at the structure of it, it contains two subunits. We call that as large subunit and small subunit. So, in the coming lessons, we will be talking about this part. So, this is responsible for protein synthesis. Okay, that's around 0.5 micrometer. Okay pa. So, what are the components? So, we have this system called as endomembrane system. Pag sinabing endomembrane system, this is a system where a particular, what do you call this? A particular set of organelles are gonna perform protein synthesis. Okay po, endomembrane system. Okay, so system of membrane-bound organelles inside the cells. So we have here nuclear envelope. Alam niyo naman yan, di ba? Endoplasmic reticulum, Golgi apparatus, lysosome, vacuoles, and plasma membrane. So, in other words, this is a typical roadmap kumbaga of where are gonna or what can we make or what proteins. Parang ito yung roadmap ng proteins class. Diba? Proteins are made up inside the cell, and after it's made up, it will then be released out of the cell for our bodily function. So rinirelease siya sa cell to perform its function then. So ito, kumbaga, ito yung roadmap, yung endomembrane system. Starting from nuclear envelope, then it will go to ER, going to GA or Golgi apparatus, lysosomes, vacuoles, and plasma membrane. Anyway, we will be talking about that here. So these components are either dalawa lang. Pwedeng continuous. Pag sinabing continuous, it's directly, okay, one system, say for example, nuclear envelope, is directly connected to ER. Magkatabi lang sila. Or, anong tawag dito? Continuous or discontinuous. Meaning, pag sinabing discontinuous, it must need a transport vehicle. Kumbaga, comparing that class in a typical environment that we if you're going to, halimbawa, magkatabi lang ng room. Pag continuous, you're going to, halimbawa, you're from one room going to another room, and that room is connected via another door in between. So you call that as continuous, meaning directly bubuksan mo lang yung door, then you are now connected to another room. Pero kapag discontinuous, you need to have a vehicles here. Say for example, you're going to another room, but then that room is actually far from where you are right now. So you will need vehicles like jeep, tricycle, or anything to actually travel into that room. Okay? So parang ganun lang din. Pag continuous, directly, okay na siya, magkatabi na siya. You're going to get from a nuclear envelope to ER. Pag discontinuous, connected siya via transfer. Kasi yung vesicles rather, is actually the transport system of the cell. Okay, so this one is the transport system of the cell. Para siyang bubble-like where the material or substance is nakasuspend sa loob ng bubble-like vesicles. Okay pa. So here, ER accounts for more than half of the total membrane in many eukaryotic cells. More than half. If you're going to look at this one, so madami kasi siya class inflow foldings. Kaya tinawag siya or accounted siya sa more than half of the total membrane in many eukaryotic cells. Okay, so ER is continuous, meaning magka-diretso lang siya with the nuclear envelope. So, sabi nga kanina, nuclear envelope, it has a continuous connection to the ER or endoplasmic reticulum. Looking at the illustration, you will see the magkatabi lang, 'di ba, yung nuclear envelope, naka-attach yung ER, right? So, it's continuous. Hindi niya na kailangan ng vesicles. There are two distinct regions of ER. Pag sinabing smooth, wala siyang mga ribosomes or bound ribosomes na naka-attach. Pag rough, meron siyang mga bound ribosomes na nasa surface. Okay, so here is the illustration showing you smooth ER. So kung makita nyo, walang ribosomes. Rough, merong ribosomes. And iba din yung structure ng smooth sa rough. Okay po? So if you're going to see this way, you're going to see that nuclear envelope is continuous to the ER. So here, in ER, we have different sections. We have ER lumen. So this is foldings in between foldings. We have the cisternae or yung sa surface ng ER. You call this one as cisternae. And on the cisternae, there are attached ribosomes, especially if it's rough ER. Okay, so we have also another name for a transitional ER. Transitional ER, it's the transition. It's the part of the ER where it's gonna transmit another molecule or protein to another organelle. Say for example, Golgi apparatus. Kasi after sa nuclear envelope, the material or the protein will be transported to ER, and into the ER, it will go to the transitional ER. The protein will be then get to the by the transport vesicle to the Golgi apparatus. Smooth ER, ano yung function niya? So, tatandaan nyo po to ha. We are talking now already of the function. Smooth ER synthesizes lipids. They create lipids, they synthesize lipids. They metabolize carbohydrates. Tandaan po class ha. They detoxifies poison. They store calcium. So, tandaan nyo po yung function. 4 yan, pakilista. Next, rough ER, sabi ko nga, meron siyang bound ribosomes. They secrete glycoproteins. Pag sinabing glycoproteins, these are proteins bonded to carbohydrates. Okay, so glycoproteins. Carbohydrates, like glycogen, and proteins. Okay, so proteins covalently bonded to. Pag sinabing covalence, di ba, sa physics, if you're going to talk about covalence, is the connection of two or more atoms. Combination of atoms, hindi siya transfer, it's the sharing of electrons in an atom. Di ba, covalence. So, pag-aralan nyo siya, maganda siya sa physics. Okay, so, di ba, we have 3 types of bonding. We have ionic bonding, we have covalence bonding, and yung isa nakalimutan ko. But then covalence, sharing of electrons. Two atoms share the same electrons. Say for example, sodium chloride. Okay, so ganun. Pag-aralan nyo na lang siya sa physics. Okay, so yan. So, ano nga yung rough ER? They secrete glycoproteins. They distribute transport vesicle. So, kaya dito, kung makikita nyo, yung vesicle or transport vesicle, galing siya sa transitional ER, which is a rough endoplasmic reticulum. Ano yung vesicles? They are proteins surrounded by membranes. Para siyang bubble-like, ito yung sabi ko. Okay, proteins are bounded by membranes for protection then. Okay. It's a membrane factory of the cell. Okay, kasi sabi ko nga, yung ER accounts for more than half of the membranes of all cells. So, siya yung nagiging membrane factory, tagagawa ng membranes. Okay, so, now that the vesicles is being transported from ER, it will now be received by Golgi apparatus. So, kung makikita nyo yung Golgi apparatus, tignan natin yung sa illustration. Again, kanina. Sa plant cell, ito yung Golgi apparatus. Okay? Meron din siyang infoldings, like folded membranes. Parang ganyan. Okay, so sa animal, yan o, kulay pink. Ito. Sana tayo. Okay, so Golgi apparatus consists of flattened membranous sac called cisternae. Yung mga surface. Function, ito tandaan nyo. Number 1, modifies products of ER. Pag sinabing modify, binabago nila, inaayos nila, parang ganun. Okay, so kaya tinawag kasi class yung Golgi apparatus as the post office, taga-receive ng information. Post office of the cell. Siya yung nagre-receive ng mga proteins transported from ER. Okay, so Golgi manufactures certain macromolecules. Diba? Yung mga macromolecules which are needed by our body. Okay, so carbohydrates, proteins, lipids, okay, nucleic acids. Okay pa. So, they also sort and packages materials into transport vesicles. So, ito po yung function niya, wag pong kakalimutan. Kumbaga, para siyang class, para siyang packaging center like yung mga J&T, yung mga sa Lazada, 'di ba? So nagpa-package siya where materials are like proteins are packaged, they are modified, then ready for transport. Na ipapadala na siya sa different branches, ipapadala sa inyo. 'Di ba? Yung mga nag-online shop dito. So parang ganoon, packaging center siya. Okay, so next, ito, Golgi body has two parts. We have, or two phases. We have cis phase. Cis phase is the receiving phase or side of the apparatus. And we have the trans phase is the shipping side. Okay? Parang ganoon nga yung sabi ko nga sa online, di ba? Meron kang mall, say for example, mag-order ka sa Lazada, say for example, or sa Shopee. Pag mag-order ka, yung seller, siya yung source. Kumbaga, yung seller, siya yung ER, yung endoplasmic reticulum. Ipapadala niya class sa packaging center. Diba? Say for example, J&T. Ano pa yung mga packaging center? Yun lang yung alam ko, J&T. So, yan, yung J&T, merong dalawang phases. Yung cis phase where it will receive the materials or the product from the ER or from the seller. Then, meron diyang trans phase. Yun na yung mag-ship. Yung J&T. Siya yung mag-ship shipping side of the Golgi apparatus. Nagigets nyo, class? Nagigets nyo?
Apo.
Okay, very good. So, you call that class as trans phase. Dalawa yung phases niya. Okay po. So, kung makikita nyo, vesicles to Golgi, it will then be packaged in a Golgi. Then, it will be shipped using again the vesicles. So, therefore, here, talking about endomembrane system, nuclear envelope to ER, it is continuous. Endoplasmic reticulum to Golgi, it is discontinuous. Okay, meaning they are just connected via vesicles. Okay pa. And also, since we are gonna use vesicles in the trans phase of the Golgi apparatus, that is already a discontinuous. Okay pa. So, yan. So, ano naman yung lysosome? So, lysosome is part of the endomembrane system. Diba? Lysosome is a membranous sac of hydrolytic. Pag sinabi natin class siya na hydrolytic, lytic or lysis, nagbe-breakdown siya ng mga water molecules. Okay po? Hydrolytic enzymes that can digest macromolecules. They can digest. So, kumbaga, ang lysosome, siya class yung parang garbage collector. Okay? Kumbaga, para siya yung dump site. Okay? So, meron siyang hydrolytic enzymes that can digest macromolecules. So, lysosomal enzymes can hydrolyze proteins, fats, polysaccharides, and nucleic acids. So, these are macromolecules. Yung proteins, fats, sugar, nucleic acids. So, some types of cell can engulf another cell by phagocytosis. This can be observed in Plasmodium, di ba? Plasmodium botulinum. They are kind of protist where they are containing tinatawag natin class yung mga false fit or pseudopodium na tinatawag. Pseudopodium is capable of phagocytosis. They engulf their food. Okay, so they obtain nutrients by engulfing another cell debris, for example. So, yan. So, phagocytosis. This forms a food vacuole. Okay, so tandaan nyo class ang vacuole, it's the storage of food, water, mineral nutrients, also wastes. Yan, so a lysosome fuses with food vacuole. Okay, vacuole is another part of a cell where it stores food, it stores water, minerals, nutrients, also wastes. And digest the molecules. Yan, nagfufuse siya, tapos dinadigest niya yung molecules by releasing the hydrolytic enzymes. So lysosomes also use enzymes to recycle the cell's own organelles and macromolecules, a process called autophagy. Autophagy. Okay? Eating of one cells. Auto meaning sarili, nagsasarili, phagy, eating. Okay po. Kinakain niya yung sarili niyang cell parts to recycle. Okay po. So yan, so kung makikita niyo class, so this is an illustration. So mag-focus na lang tayo dito sa digitalized illustration, mas maganda. So we have A and B. So let's talk about A, which is phagocytosis. If you're going to get this as this is the food, say for example, the food will be then transported from the plasma membrane, then yan, so via vesicle. Then here comes the lysosome where it contains hydrolytic enzymes. It will then fuse to the food vacuole. Okay, and the enzymes released by the lysosomes will digest the food in a vacuum. Okay, in a process called digestion na tinatawag. Okay po, so this is a form of phagocytosis. Okay po. So ganyan. So next we have autophagy. 'Di ba? Pag sinabing autophagy, recycling one's own organelles. So here is a peroxisome of ano nga yung peroxisome? It's present in, looking at the parts, peroxisome, yan. So present siya sa plant cell. Okay, yung mga ganyang itsura. Ito. Yan. Peroxisome. Meron din siya sorry. Meron din siya sa animal cell. Yan, mga peroxisome. Layo na natin Yan. So, peroxisome, okay, is inside, or this is the non-functional peroxisome and a mitochondrion enclosed by the vesicle. The lysosome reaches the vacuole, okay, that contains mitochondrion and unfunctional peroxisome and mitochondrion. Then it will digest. Okay po. So, parang ganun. Autophagy. Digestion of one's organelles, cell's organelles. A plant cell or fungal cell may have one or several vacuoles. Plant and fungal cells. Especially, very evident siya class sa plant because a plant contains one large vacuoles that comprises most of the cell's internal space. Food vacuoles are formed by phagocytosis. So, we have this term class as contractile vacuoles. Contractile vacuoles, from the word itself, it contracts. This can be found among protists. They pump excess water out of cells. So, kaya nga, contractile vacuoles. Okay pa? So, central vacuoles, this can be found among matured plants. They hold organic compounds and water. Okay po, central vacuoles and contractile vacuoles. Two types of vacuoles. Okay, ito yung sa plant cell. Meron siyang central. Pag sinabing central kasi, parang malaki. Okay, siya yung nag-occupy aside from the nucleus, occupies greater space inside the plant cell. Okay, so ganyan. Endomembrane system, tandaan, it's a complex and dynamic player in the cell's compartmental organization. Tandaan nyo yung endomembrane system, ito, balikan natin. So, lysosomes, vacuoles, then plasma membrane. Okay po. So, they are connected via vesicles. Okay, and dynamic player in the cell's compartmental organization. So here is the illustration of endomembrane system. Diba? Starting from nuclear envelope, it's connected continuity. It's connected directly to rough ER, and then the rough ER is gonna yung rough ER, meron siyang tinatawag nating ano nga yun, yung transfers where it ships the molecules or the proteins via vesicles to the this phase of Golgi. The Golgi will then transport, will then package, sinosort yung material. Okay, pagdating sa phase ng Golgi, it will ship the packaged material, okay, to the plasma membrane, and plasma membrane will receive the material, then it will be released to the environment or to the outside cell. Ganyan po yung itsura niya. Ano naman yung mitochondria? So mitochondria is commonly known by many of you as powerhouse of the cell or energy generating organelle. Okay po. They are sites of cellular respiration where it generates ATP or the energy we use for our daily living. Diba? So without energy, we might not do bodily processes. Okay, so we have chloroplasts. This is intended for plants capable of photosynthesis. So they are also found in algae. They are sites of photosynthesis. Also, energy generating organelle in plants. Peroxisomes are oxidative organelles. Okay, so mitochondria and chloroplasts are not part of endomembrane. Okay, so they are not part of endomembrane kasi ano yun lang, ang mitochondria and chloroplast, they are just capable, or they just create energy source needed by the cell. So they are also double membrane. They have proteins made by free ribosomes, and they contain their own DNA. So this explains the endosymbiosis of the history of life. Okay, but then that's not our topic for today. Okay po, so tandaan po. So mitochondria are nearly all eukaryotic cells. Wala ka naman makikita sa prokaryotic na mito. Smooth outer membrane and inner membrane. Okay, inner membrane folded into cristae. Hindi siya cisternae. Okay pa. The inner membrane creates two compartments. Okay, so meron tayong intermembrane space and mitochondrial matrix. Okay, so makikita natin yan in a picture. Okay, so kung makikita nyo, ito yung inner membrane. Okay, so intermembrane space and mitochondrial matrix. So ito, meron siyang double layer, right? So between the second layer and first layer, you will call the space as intermembrane space. Yung matrix naman, it's the space between the second membrane or the cristae or the infoldings. Okay pa? So Yan. Some metabolic steps of cellular respiration are catalyzed into mitochondrial matrix. Okay, so these crystals present a large surface area for enzymes that synthesize ATP. Okay, but then we will not talk about cellular respiration and cell respiration and photosynthesis in this topic. Okay, so more on overview tayo ng cell parts. Chloroplast is a member of family of organelles called plastids. They contain green pigment called chlorophyll as well as enzymes and other molecules in function of photosynthesis. Kung yung mitochondria, it's for cellular respiration. Yung chloroplast, it's for photosynthesis. So they are found in leaves and other green organs of plants and algae. Chloroplast, meron din siyang internal structures like mitochondria. We have thylakoids, membranous sacs stuck to form a granum. And stroma, it's the fluid inside the chloroplast. So kung makikita nyo, ito yung ribosome, ito yung chloroplast, whole chloroplast, meron siyang mga attached ribosomes. Thylakoid, para siya mga pisong pinagpatong-patong. You call that class as thylakoid. And the fluid inside the chloroplast, you call that class as stroma. Okay? So also, chloroplast is a double membrane. Okay? So, an organelle for photosynthesis. So, ano naman yung peroxisome? Peroxisome are specialized metabolic compartments bounded by a single membrane. Okay, so compartments din siya. Okay, pero single membrane compared to chloroplast and mitochondria. They produce what? Hydrogen peroxide and convert it to water. Okay? So, kumbaga, ano siya, water source yung peroxisome. Oxygen is used to break down different types of molecules. So, very important yung sa kasi baga yung peroxisome, it's the source of oxidation process. So, meaning, presence of oxygen which help break down different types of molecules. Yan. Yan yung peroxisome. This is a section of the cell where peroxisome is present. Single layer lang siya compared to a chloroplast na dalawa and mitochondria. Pero, in this illustration, mas malaki yung peroxisome sa mitochondria. Okay, what about cytoskeleton? Cytoskeleton is a network of fibers, kaya nga siya parang skeleton din, extending throughout the cytoplasm. Cytoskeleton is a form of protection, a form of structural support of the cell. Parang in our body, we have bones. In a cell, we have cytoskeleton. So it organizes the cell structures and activities anchoring many organelles. So it's an anchor for an organelles, yung cytoskeleton. Composed of 3 types of structures. We have microtubules, which are bigger among all the structure of cytoskeleton. Microfilaments, the smallest ones, para siyang mga sinulid. Intermediate between the two. So, kung makikita nyo class, ito yung mga microtubules which are larger, microfilaments which are smallest. Okay, support and maintains its shape. So, it also interacts with motor proteins to produce motility. Diba? Yung sa contraction. Inside the cell, vesicles can travel along monorails. Pag sinabing monorails, one path lang siya provided by cytoskeleton. Recent evidence suggests that cytoskeleton may help regulate biochemical activity. So here is a typical cytoskeleton interacting with a particular motor protein. Okay, so yan. So, 'di ba yung vesicles yung vesicles in an endomembrane system is transported from ER to Golgi to plasma membrane. It won't be transported without the help of motor protein and cytoskeleton. So here, in the transport of vesicles, we need sorry. Inuubo kasi ako. Yan. So, we need the help of motor protein and cytoskeleton in the transport of vesicles. Okay, so, yan. We need energy to actually actuate that. Okay, so, here, we have the receptor for motor protein. So, parang receptor where vesicles can attach itself. And we have ATP. And we have here the motor protein. Motor protein, ATP powered. Parang kumbaga siya yung naka-attach dun sa microtubule ng cytoskeleton in order for it to move from one location to another. Okay, so 3 main types of fibers that make up the skeleton. We have microtubules, filaments, and intermediate. Intermediate between microtubules and microfilaments. Thickest of them all is microtubules. Okay, so microfilaments is the thinnest of them all. So they are called as the actin filaments. So intermediate, diameters in the middle range. So yeah. Yeah, so microtubules, so they are hollow, consist of 13 columns of tubulin molecules. So, ito yung data class. So, paki-screenshot na lang din. Ito na microtubules, ito. They are called as tubulin polymers. Ito naman, microfilaments, hindi siya tube, hindi siya hollow, but they are made up of 2 intertwined strands of actin, parang ganito. Yan. Yan, ganyan yung itsura niya. Intermediate, parang ganito. So, somewhere in between. So, it's fibrous protein super coiled into thicker cables. Para siyang mga cables like this. Diba? So, ganyan yung itsura niya. So, yung functions niya, microtubules, maintenance of cell shape, cell motility, chromosome movements in cell division, and organelle movements. Main function ng filaments, maintenance of cell shape, parehas, changes in cell shape, muscle contraction kasi it's made up from actin filaments, cytoplasm streaming, cell motility, and cell division. Cell division is actin microfilaments is present during cell division. Intermediate, maintenance of cell shape, anchorage of cell nucleus and certain other organelles, and formation of nuclear lamina. So, ito yung nagko-compose ng nuclear lamina. So, yan. So, ito na yung functions which I already stated. Shaping, movement, separating chromosomes during cell division. So we have another parts of a cell. We have centrosomes and centrioles. Centrosome is where microtubules grow out, so it's near the nucleus of a cell. It's also called as the microtubule organizing center. Para siyang center, para siyang location where microtubules are organized din. In animal cells, the centrosome has a pair of centrioles. So, dalawang centrioles. Okay? So, each with 9 triplets of microtubules. So, 9 triplets of microtubules arranged in a ring. So, if you're going to look, so this is another extra task. So, pag-aralan nyo yung sa cell division ng cell. If you're going to look at that, at that particular parts of the cell, yung microtubules, you'll see kung ano yung kanyang structure. It's a 9 triplets. So, parang ganito. 9 triplets na microtubules. Ito yung buong centrosome. Sabi ko nga, it's the organization of microtubules. Centrosome. Centrioles yung dalawa. Okay? Pag sinabing centrioles, it has it's a pair, okay, of microtubules. A pair of microtubules which are triplets, 9 triplets of microtubules. So kung makikita nyo, if you're going to count the triplets, 1, 2, 3, 4, 5, 6, 7, 8, 9. So it's made up of 9 triplets of microtubules arranged in a ring. So parang ganito siya. Okay, so yan. So this is the longitudinal microtubules. Okay, so yan. Centrosomes, centrioles, and microtubules are very important in cell division. Okay, so cilia and flagella are both for movement. So they are called class as locomotor appendages of cells. Okay, so di ba yung sa sperm cell contain a flagellum. Very important for propelling the sperm cell, di ba? So kung makikita nyo yung sperm cell, meron siyang flagellum. Yung cilia naman, sabi ko nga, present siya sa small intestine natin, present siya sa linings ng ating tube papunta sa lungs. It's responsible, kung sa intestine, responsible siya for absorption of nutrients. Okay, so kasi diyan naka-rely yung mga diyan nakalagay yung mga absorbing mechanisms ng nutrients sa cilia. So, yan yung kung sa kung sa linings naman ng ating sa trachea naman natin, meron yang cilia where kapag may dust particles, may foreign objects, so umuubo ka, so yung ubo mo with the help of cilia, it expels out these foreign objects. So, they are different, yung cilia and flagella, in beating patterns. So, kung makikita nyo, yan, direction of the swimming is going horizontally, but then yung beating patterns niya, it's diagonally. Motion of the flagella. So, yan. So, direction of the organism's movement, ito naman, power stroke niya, ganyan. Power stroke, para siyang catapult, right? So, catapult, tapos, yan. So, meron tayong recovery stroke. Power stroke, meaning magbibuelo. Then, recovery stroke, that's where it will move the organism. Parang ang catapult ba? So, mas maliit yung mas maliit yung flagella as compared to the cilia or cilium. Okay, so whenever kahit na sila ay magkaiba, so they still share a common structure. The core of microtubules sheeted by plasma membrane. The basal body of that anchors the cilium or flagellum. So ito yung pagkakaparehas. A motor protein called dynein which drives the bending movements of a cilium or flagellum. So ito yung motor proteins. So tinatawag natin na dynein. So we have actin, we have dynein. Yan yung mga motor proteins. So pag-aralan nyo siya. So yan. Kung makikita nyo, so this is a structure of a typical cilium. Yan, yung basal body niya, so parang yan, 9 triplets. 9 triplets. So, ganyan yung itsura niya. Okay, so sa base niya, meron siya dynein structure. So, yan. How dynein walking moves flagella and cilia. Dynein arms alternately grab, move, and release the outer microtubules. Protein cross-links limit sliding, and forces exerted by dynein arms cause doublets to curve, bending the cilium or flagellum. So, in this case, so hindi nyo siya talagang mas maiintindihan kung babasahin ko lang sa inyo how dynein works. I want you to watch na lang sa YouTube. Madaming mga animated videos search nyo how dynein works. Okay po? Para at least makita nyo kung paano ba gumagalaw yung flagellum or cilia. Okay, so let's skip this. Okay, microfilaments. So, okay na yon. Okay, so ito yung sinabi ko sa inyo kanina, yung pseudopodia or the false feet. Relatively present among protists where they are capable of phagocytosis. Okay? Pseudopodia are called as the cellular extensions. They are not actually feet, kaya nga siya pseudo, false, podia, feet, false feet. They are cellular extensions that extend and contract through reversible assembly and contraction of actin subunits into microfilaments. Present in amoeba, okay, these are actually protists. Cytoplasmic streaming is a circular flow of cytoplasm within cells. So, meron tayong, in a cytoplasm, we have this term as cytoplasmic streaming. Ang cytoplasmic streaming, they speed up it speeds up the distribution of materials within the cell. In plant cell, actin-myosin interactions and sol-gel transformation drive cytoplasmic streaming. Okay, so ito na yung function ng intermediate filaments. Okay, so we have these extracellular components of the cell, or sometimes they are mostly external to the plasma membrane. There are members of the kingdom, say for example plant and protist and bacterium, where there are called as extracellular structure, like cell wall in plants for protection, for structure of plant cell. Kaya 'di ba, yung plant cell can withstand higher temperature sa tanghali. Okay po. So because of cell wall. Unlike with animals like us, hindi natin kaya magbilad maghapon. So we have also extracellular matrix or ECM of animal cells and intercellular junction. Cell wall structure that distinguishes plant and animal cell. But hindi lang sa plant ang protists ay ang bacteria, prokaryotes meron ding cell wall. Diba yung pinakita ko sa inyo kanina? Prokaryotes, fungi meron din, yung mga plant-like fungi, ay yung mga fungi, meron ding cell wall, and some protists, yung mga plant-like protists pala. Okay? Protects, maintains the shape, prevents excessive uptake of water. They are made up of cellulose fibers. Okay, embedded in other yung tinatawag natin na glycoproteins. Okay po. So polysaccharides and protein. So yan. So plants may have multiple layers. We have primary cell wall, relatively thin and flexible. Middle lamella, thin layer between primary walls adjacent cells. We have secondary cell wall added between the plasma membrane and primary cell wall. So, yung plasmodesmata in plants are channels between adjacent plant cells. Kung sa animal cells, we have yung mga embedded proteins. So, sa plant naman, meron tayong plasmodesmata. Ito yung kumbaga channels. So, ito, secondary, primary, and middle lamella. So ito is one cell, another cell, middle lamella, it's between, okay, primary walls of adjacent cells. Okay, so primary we have middle lamella, and we have secondary, yung sa loob or sa ilalim ng primary wall, and this is connected via plasmodesmata, yung mga pores in between. Okay pa. Okay, ECM lack cell walls but are covered by ECM. Okay? So, animal cells, wala silang cell walls, pero meron silang ECM. ECM is made up of glycoproteins, sugar, and proteins like collagen, proteoglycans, fibronectin. ECM proteins bind to receptor proteins in the plasma membrane called integrins. Okay, so integrins. So, yan. Collagen, fibronectin, yung mga proteins. So, ito yung integrin na kanina. Yung protein where carbohydrate chain are attached or is attached. Ito. So, paki-screenshot po in order for you to study it later. Yeah. Functions of ECM: support, adhesion, movement, and regulation. For animal cells lang yan. Okay? So, intercellular junctions. Ito, we have plasmodesmata, tight junction, desmodomes, and gump junctions. Pag sinabing intercellular, this connects one cell to the other cell. Okay? Or you call that class as surrounding or neighboring cells. They communicate, they are connected via direct physical contact. Okay po. So, dapat ang one cell must coordinate to neighboring or surrounding cell to actually perform the function of tissues, organs, organ system, 'di ba? Yung pinag-usapan natin yung sa ano tawag dito? Pinag-usapan natin yung sa levels of biological organization. A tissue is composed of group of similar cells which coordinate, dapat nag-coordinate sila via junction, intercellular junction, to perform a particular task. Same thing with organs. Organ is a group of tissues where it should be communicated. Cells need to be communicated to perform its function. Okay. Plasmodesmata, channels in the cell walls. Yan. Water and small solutes and sometimes proteins and RNA can pass from cell to cell. Anyway, so we still have 8 minutes. So, yan. So, yan yung tatlo. Tight junction. So, membranes of neighboring cells are pressed together, preventing leakage of extracellular fluid. Okay, they are pressed together. Desmodomes or anchoring junction fasten cells together into strong sheets. Gap junctions, communicating junctions, provide cytoplasmic channels between adjacent cells. So I want you to take note this one. I'll give you an extra task to watch the animation in YouTube. So search nyo lang yung different intercellular junctions of the cell para at least you will be aware of what are these three ano yung function nila for cell. Okay, so yan yung tatlo. Okay, so ito na yung sabi ko. Cell rely on integration of structure and organelles in order to function. Dapat they should coordinate. Okay, organelles should coordinate to each other to function as a whole. Or kaya nga, a living unit cell is greater than the sum of its part. Okay po? So, for example, a macrophage ability to destroy bacteria involves the whole cell coordinating components such as cytoskeleton, lysosome, and plasma membrane. If one of these fails, meaning the function of the cell or the mission of the cell will not be will not be done. Okay po? Kasi nga, one is malfunctions. Dapat they should coordinate to each other. So, like that one. Okay? Anyway, I'll be giving you ito yung sa microtubules. Okay, so yan. that's gonna be the end of my discussion. So I hope you learned something. Wait for a while.